I'd also like to point out that in addition to myself, my colleague Victoria Smith will also be here to answer questions as well as Tim. All right. So the first thing I'd like to just talk to you about as an introduction is just our general concept of how you would advance a biomarker candidate from discovery through qualification. And I think one of the first things to just make sure we're all on the same page about is the definitions of things like biomarker, context of use, and fit for purpose. A biomarker is an indicator of a normal or pathological process or of a response to a therapeutic. A context of use is the manner and purpose of use of the biomarker. In other words, things like the type of biomarker, i.e., is it a diagnostic biomarker, is it a prognostic biomarker? And also, context of use is an important thing to think about in terms of the modality of the biomarker that you're using or exactly how it will be used. Fit for purpose refers to the degree of validation that you need, and of course that's dependent, again, on what you want to do with your biomarker, what your plans are for that biomarker. So in this particular diagram, you see the top bar shows how you're going from activities that are primarily in the laboratory into the clinical setting. And the chevron shows various aspects of this process, such as discovery, validation, clinical utility, and qualification. And you can see below that one thing that changes as you move from discovery to qualification is the degree of evidence that's required to show that your biomarker is robust, reliable, and actually does what you want it to. So some of the activities that are done during discovery include finding a sample source for your potential biomarker that has standardization as a requirement, that has annotation and so on, so that you actually are, again, you're getting what you look for. Once you have your sample source and you're starting to use it to identify your biomarker, you of course need bioinformatics. So usually bioinformatics and statistics are a very important part of discovery. And then another aspect, in addition to identifying the biomarker and doing some initial proof of concept work with it, is to begin defining your context of use. You'll find throughout this whole process, understanding your context of use and defining it becomes one of the most important aspects of figuring out exactly what your validation scheme is and whether you want to go for qualification of your biomarker with the FDA. In validation, context of use, again, further definition of it is important. And here, so is analytical validation. And we'll be talking about that quite a bit during this little presentation. Basically, analytical validation has to do with establishing the performance characteristics of your biomarker measure. There's also clinical validation that occurs, and that's the process whereby you correlate the biomarker with a clinical concept of interest. When you're defining clinical utility, this is when things get extremely expensive. I think another thing you can see, in addition to the degree of evidence required as you move from discovery to qualification, the expense also increases. So in clinical utility, usually these are large prospective clinical studies, again with a very defined context of use. The purpose of these are to evaluate the overall utility for clinical practice and for drug development. Finally, when you reach the level of qualification, we'll talk about this a little bit more as we go through the presentation, but this is an FDA review that's required for that. They have an entire program devoted to the process of qualification, which we'll talk about a little bit. And then within the context of use, once you have qualification, the biomarker should provide a reliable application and it should allow specific interpretation of what your biomarker is telling you. Okay, move on to the next one. So I'd like to spend just a little bit of time talking about our Neuroscience Biomarker program as it is right now. So the goal of the program is to facilitate the development of high-quality biomarkers to improve the quality and efficiency of clinical research in phase 2 and beyond. We have 3 basic pillars to this program as we've set it up right now. The first is about NINDS funding opportunities, which we're going to focus this presentation on. These will support a rigorous fit-for-purpose validation process. Notice of interest, encouraging applications focused on pain biomarkers actually is in the queue right now for publication. So this is one thing we'll talk about a little bit throughout the presentation as well, but right now there is really an emphasis on finding pain biomarkers and validating them. So we are encouraging people who have an interest in that type of research to apply for these particular funding opportunities. Another pillar is a workshop focused on best practices in neuroscience biomarker discovery. This is planned now for late 2018, most likely in reality early 2019. And then the last part of the plan is a centralized NINDS one-stop webpage that will have links to NINDS biomarker resources, including some of the repositories, as well as information from the FDA and public-private partnership opportunities. Okay. So this slide tells you, that I'm on the third slide right now, that tells you about the funding opportunities supporting a fit-for-purpose validation process. So we have 4 funding opportunities, but really we only have 2 in concept. We have one for analytical validation of a candidate biomarker for neurological disease. We have a U01 version of that, as well as a small business SBIR version of that, which is the U44. We also have a funding opportunity for clinical validation of a candidate biomarker for neurological disease. Again, the plan is to have a U01 version and an SBIR version. The SBIR version is published, as are the 2 analytical validation funding opportunities, but the clinical validation U01 version is still waiting in the queue for publication. It should be coming extremely soon. We have all the information on it already, so just stay tuned for that one. And then just so you know, for the 3 that are already published, the application due date is March 14th, 2018. All right. So to talk a little bit then about biomarker definitions, just so we're on the same page about what analytical validation is versus clinical validation, we define analytical validation as establishing that the performance characteristics of a measurement are acceptable in terms of sensitivity, specificity, accuracy, precision, and other relevant characteristics using a specified technical protocol. And that technical protocol can include sample collection and standardization procedures. The other thing that's important in understanding analytical validation is the level of analytical rigor that is necessary depends on the characteristics of the biomarker, the detection technology, the type of clinical question, or the intended use as a biomarker, i.e., is it a diagnostic, predictive, pharmacodynamic biomarker, and so on. So in terms of defining clinical validation, that's establishing that the biomarker acceptably identifies, measures, or predicts the concept of interest. And then finally, clinical utility is defined as the conclusion that the given use of a biomarker will lead to a net improvement in health outcome or provide useful information about diagnosis, treatment, management, or prevention of disease. We are getting these definitions from a resource called Biomarkers, Endpoints, and Other Tool Resources. This resource was compiled by a collaboration between the NIH and the FDA, and you can look at it as sort of a living document. We have the link that on these slides, and the link is live. But in addition to that, you should actually just keep up with it every now and then, check it, because things change with that. Now for the next slide, I'd like to go through the cooperative agreement mechanisms and exactly what they involve. So both the U01 and U44, i.e. the academic and the small business versions of these, both of them are cooperative agreements. They both have a potential for 5-year durations with go-no-go milestones after each year. One thing that's important to remember about the go-no-go milestones is that we mean it when we say go-no-go. So the idea for the milestones is that they represent decision points regarding whether or not it actually makes sense or is feasible to go on to the next year of funding. These milestones should be in your applications, and then if you get an award, we, i.e. Victoria, myself, and the program officer from the applicable disease area, work with you to help finalize the milestones. We want to make sure that they are realistic, and we want to make sure that they actually provide decision-making opportunities. And so we need to see that they have quantitative criteria for success supporting each of them. So once we go through this process, you finalize your milestones and you get to after your first year of funding and you're actually in this administrative review, we work with you to try to come to a decision about whether or not you've met your milestones. If you do not meet your milestones, you won't be funded for the next year or for the rest of the grant, basically. We try to make that a very rare circumstance. So it should be a case whereby you know whether you're going to meet your milestones or not, and if you have any questions throughout the year and you want to have a call with us, we want to work with you. And we're in the process right now, too, of trying to assemble a team that consists not only of those of us that I've just mentioned, but also perhaps any experts that are internal that can provide advice on statistical analyses, experimental design, any technical issues that may come up. So, and also the FDA is working with us on this too. So we hope we have a team that can help you through this entire process. And as I said, transition occurs via this administrative review, and the opportunity for transition occurs once a year. All right. So the next slide just gives you a nice table that summarizes the mechanism itself, the name of the mechanism, the length, and then the budget that's associated with the mechanism. So for the U01, probably, I don't know, a lot of you might be familiar with this. The length for these particular funding opportunities are up to 5 years. Budget is not limited, but it has to reflect the actual needs of the project, and of course that's a judgment that's made as we prepare to fund. In terms of the small business version, the U44, phase 1 is up to 2 years, phase 2 is up to 3 years. Phase 1 is up to $700,000 per year. Phase 2 is up to $1.5 million per year. But again, must be reasonable and appropriate. And again, it's something that is a decision point that we make as we're about to fund. Okay. So now I'd like to go through each PAR. First of all, the analytical validation PAR, and of course I won't do anything separately for the SBIR version. But the goal of the analytical validation funding opportunity is to encourage rigorous analytical validation of a candidate biomarker or endpoints in a manner that is consistent with FDA guidelines. Things like evaluation of the assay, its performance characteristics, and optimal conditions are part of these guidelines. To facilitate, the other goal is to facilitate the advancement of robust and reliable biomarkers to the application in clinical trials and practice. So the idea is that when you're finished with this, you would have a biomarker that is validated to the extent that it can be used reliably for whatever your context of use and fit for purpose idea is. We want to emphasize that, you know, phase 2 clinical trials and beyond are really where it appears they're needed. Biomarkers make a huge difference in the success of moving from phase 2 to phase 3 and from phase 3 to nomination for new drug application. Entry criteria for the analytical validation funding opportunity include that you have an identified biomarker with a working hypothesis on the context of use. So this brings to mind one thing. We want to emphasize that these funding opportunities are not meant to do the initial identification and discovery work on a biomarker. We are assuming you've identified a biomarker that you can actually call a candidate. The other thing this entry criteria requires is that you have a developed method of detection. This is not for technology development either. We do understand, though, that even though you might have a method developed already, you're probably going to need to optimize it throughout this funding opportunity, and that's absolutely okay with the funding opportunity. Another entry criteria are plans for management of pre-analytic variables, and these pre-analytic variables include things, again, like making sure that the sample collection that you've based your biomarker selection on was standardized, that sample sources were annotated, and so on. That has to be something that's made, actually, when you're writing your introduction, it will be important to show that you've taken these things into account, and it will be important to show that you have evidence that you are working from standardized samples. Another entry criteria is that you have a cogent biological rationale supporting the candidate biomarker, and that your biomarker has relevance to therapy development or clinical practice. Examples of appropriate activities during this, the analytical validation FOA are, you could be doing things like determining the context of use or further refining it. You could be doing analytical validation of a tissue or biofluid, imaging, physiological or behavioral biomarker assay. So as you can see from this particular example, we really don't care exactly what modality you're using for your biomarker. It can be anything from tissue to imaging to behavioral. You could be doing analytical validation of biomarkers with a clinical intervention, and you might need to do this even if you're just doing analytical validation because you need to see what your dynamic range was, for instance, for whatever therapeutic you're using in the case of pharmacodynamic or response biomarkers. You could also be doing studies involving analytical validation of biomarkers without clinical interventions, such as diagnostic, prognostic, or susceptibility risk biomarkers. Another example of appropriate studies are those focusing on improving the standardization or harmonization of assays among laboratories for use in clinical trials. Imaging comes to mind right away as something where the need for standardization is very important, and if that's part of your application, that certainly is relevant to analytical validation. The goal for analytical validation should be that the biomarker measurement meets FDA analytical performance criteria. And again, as Tim said, you'll be sent these slides, and the link is live, so you can take a look at those guidances. Examples of things that are out of scope for the analytical validation funding opportunity are natural history studies, aid in understanding disease, biomarker identification itself, initial development of biomarker detection methods. Again, although we expect that you will have to continue to optimize your detection methods as you're doing your validation. Therapeutic target identification is out of scope. Preclinical animal studies are out of scope. Development of candidate therapeutics are out of scope. And studies focused on clinical validation are out of scope because our other funding opportunity covers that. And I think, as we've mentioned before, analytical validation can include metrics such as characterizing accuracy, precision, sensitivity, reportable range of test results, reference intervals, harmonization of performance, and any other performance characteristic required for test performance with determination of calibration and control procedures. And what's in italics here is applicants with assays that may have already met the above criteria could go ahead and directly apply to the companion funding opportunity, which is the clinical validation funding opportunity. So now let me just run through some of the same things to address on the clinical validation of a candidate biomarker. The goals of that one are to encourage rigorous clinical validation of a candidate biomarker using retrospective and or prospective methods in a manner that's consistent with the purpose of the biomarker. Another goal is to facilitate the advancement of robust and reliable biomarkers to application in clinical trials and practice again. Entry criteria for the clinical validation funding opportunity are that you have identified the biomarker and have a working hypothesis on the context of use. Another criteria is that the analytical validation of the biomarker should be completed and it should be consistent with FDA standards. So if you apply to the clinical validation funding opportunity, we are assuming that you have evidence that you've already done analytical validation for the measure itself. Another entry criteria is that there are plans for management of pre-analytic variables, which we've talked about before. And again, this has to do with your sample sources and standardization. We also assume that you have preliminary validation criteria or data, and it should be presented in your application. And again, you need a cogent biological rationale, and you need to show relevance to therapy development. Some examples of clinical validation studies that you might be doing while being funded through this opportunity are clinical validation studies of the biomarker with the goal of establishing robust correlations between biomarker and disease or therapeutic response. And then we are assuming, as I stated before, that you might still be doing some optimization of your analytical validation. You could also be expanding your determination of context of use, because we also assume the more you learn about your biomarker and how it correlates with the clinical conditions that you're interested in, you might have to make some changes in your context of use document, or you might even really expand the scope of it. Where you could also be doing retrospective, well-controlled multi-site clinical studies. In addition, you could be doing prospective studies. And again, this just depends on what you want, what the biomarker is and what you want to do with the biomarker. You might also be doing studies designed to assess the health impact of a biomarker in clinical practice, among others. I'm not going to read all of the ones on this slide. Right, some examples of things that would be out of scope for the clinical validation opportunity are, again, natural history studies aimed at exploring disease pathophysiology. Applications that propose any animal studies are out of scope. Studies of biomarker identification or technology development, again, are out of scope. If analytical validation is the sole intent, that's out of scope for obvious reasons. Applications that propose only to create or maintain patient registries are not in scope. And again, you cannot be using this funding opportunity to test a candidate therapeutic. And clinical intervention studies, other than those necessary to validate the biomarker, are out of scope. And then applications that request support for infrastructure to establish new networks are also out of scope. Clinical validation can include the following types of metrics: demonstration of an association of the results of the biomarker with a clinical endpoint, definition of the sensitivity and specificity of the assay result within the context of defined clinical endpoints and clinical population. In other words, receiver-operator characteristic curve development certainly is part of the activities you'd be doing in this funding opportunity. And then you could also be estimating the prevalence of the marker within subjects or patients for the intended clinical context. And then you could be establishing appropriate cutoffs or thresholds for the assay. Another thing that we really encourage for either the analytical or the clinical validation funding opportunity is that you set up collaborations that are multidisciplinary. We understand that developing biomarkers is a very complex thing to do, probably one of the reasons that there aren't very many well-validated biomarkers in the neuroscience space. So we encourage you to have a team that includes clinicians, people on the technical side for assay development, statisticians, consultants that you need, clinical laboratory staff, and if you have individuals that are knowledgeable in the FDA qualification process, that is good as well. And as I said, we are going to be trying to work with the FDA so that if we need their advice or expertise for your particular project, we can draw them in. Okay, the other thing that's important to remember is that we really do encourage you to leverage existing research resources within NINDS and the NIH. We've listed some examples of research resources that you could draw from, and most of these have to do with your initial discovery in the first place, where you can get sources of human tissue, imaging data, and just overall bioinformatics data on these sources. There are also resources that can be found from, of course, private research foundations and other government agencies. Okay. So this last slide just goes through a little bit about the qualification of biomarkers. One thing, if qualification is the reason that you would like to be funded through these opportunities, then we really encourage you to contact the FDA even before you send in the application or as you are sending in the application, so that they can work with you step by step as you're being funded by the opportunity. Biomarker qualification is really a process where you build the evidence. If you remember the chevron in the first slide, where you build all of that evidence you need to support using your biomarker for clinical practice or for clinical trials. And as you build this evidence, the whole process of qualification, what they do is they set up a team to work with you at the FDA, and they have also sort of like milestone points where you look at the evidence that you have at a certain point, the team looks at your evidence, and they decide whether or not you can move to the next step. So as I said, they work with you step by step until you finally reach the point where the FDA committees believe that they can classify your biomarker as qualified. And that should actually go a long way in terms of, allowing multiple clinical trials to make use of your biomarker. It could even put you on this pathway to commercialization. All right, so I think that's pretty much all I've got. So we could start working on the questions, right? Oh, before we start on the questions, I just want to point out the last slide gives contact information and resources. So please take a look at these slides. Again, all the links are live. There are links to the funding opportunities, both the analytical validation and the clinical validation, as well as an initial website that will basically go over what I just did in this presentation. So please feel free to look at all of those things. All right, so now I think we'll start with the questions that were sent to Tim before I started the presentation, and then I'll just keep moving through things in order. So the first question is, would proposing to use diffusion tensor imaging and MRI to validate a fluid biomarker be responsive to one or more of these RFAs? And so I would emphasize here, again, we were really pretty much agnostic when it comes to the modality of the biomarker or the type of biomarker. We just need to see that it has use for neurological disease. So if you're trying to figure out which funding opportunity to use, I guess you have to ask the question, is the main goal to do analytical validation of that biomarker, or is it to do clinical validation? And that should help you decide which one to go for. And of course, you can call us. We're more than happy to schedule teleconferences with you if you're still trying to figure out which one to apply for or if you should apply. All right? So the second question is, does NINDS have programmatic interest in clinical validation saliva-based biomarkers for diagnostics, prognostics of TBI? And the answer is yes, we do. So again, that seems like a very interesting thing to do. So again, figure out whether you look at the funding opportunity just to make sure that you have what it would take to meet the entry criteria. And then if you have questions, call us. The third question is, can adult as well as pediatric patients be included in the clinical studies? And the answer is yes. The fourth question is, besides the fast track, is there also direct to phase 2 mechanisms? And I'm assuming when you're asking us about this, you mean the SBIR versions. And the answer there is, I believe that you cannot use direct to phase 2. I think you have to use both phases, and that's primarily because it's a cooperative agreement mechanism to the U44. So, but I gotta say I'm not completely sure, so I will find that out for you. So please email me that question as well. And then the question is LOI, letter of interest due today. And the letter of interest, I should stress, is not absolutely required, but if you are going to send one, it would be great if you send it today or as soon as possible. Okay. Besides March 14th, there are later due dates, correct? And that is true. There are later due dates, and I believe if you look at the links, it should tell you what the other due dates are, or if they are I think they are not standard due dates, so it should tell you. And for the fourth funding opportunity, which isn't published yet, we have information to lead us to believe that the due date on that one will be in mid-April, but you know, we can't say anything for sure until it's actually out there published. And so remember, that one that's not out there is the U01 version of the clinical validation. So the next question is, in general, can any prognostic biomarker for TBI be submitted for this program? And the answer is yes. Again, we're not going to, you know, be specific about what types of biomarkers you can do. We're more interested in helping you through the validation process. And in terms of what would be of more interest, probably for questions like that, you should contact us, and we'll get our disease area program official to be in on the call with us, and they might be more able to help you answer the question of what is of more interest. Okay, then just to clarify, does the U44 include both phase 1 and phase 2? And yes, it does. And then does the NINDS support vision-related applications? I think if it has to do with the retina, they do, but that's another one of those questions where I would encourage you to email me, and I will get the program officer who's relevant to this disease indication to help me answer that question for you, and we could even have a call if you would like. Okay, are others of interest, such as biomarkers related to large vessel occlusion? Biomarkers related to large vessel occlusion are definitely of interest, as long as it's in the brain. Okay. Next question is, it seems to be putting the cart before the horse to have all these grants funding analytical validation of biomarkers first, when the real issue for most neurological biomarkers is a lack of translation to humans from animals, not validation. We do agree with your assessment there. Part of this opportunity and this whole plan that we have is actually a bit of an experiment to see what types of candidate biomarkers actually come in for our validation funding opportunities. But in the meantime, I mean, we're hoping actually that there are some candidate biomarkers that look good that come in. But in the meantime, that's one of the reasons we also want to have the workshop, because that one will be focused on how do we do a better job in discovering biomarkers that are translational. Now, can you please help to find the FDA standards for biomarkers? So the FDA does not have like a guidance that just is entitled the way you've posed that question. There are guidances that relate to analytical standards, and I think that's in the funding opportunity description. So if you click on that link, you'll find it. When it comes to clinical validation, there are some standards, but there's not a specific guidance on that. And so we have put together our standards, some of the things that I listed, for instance, in this presentation, from a combination of looking at guidances from the FDA and looking at review articles that are current. And again, so if you'd like to discuss this more, please feel free to email me. Is there an anticipated funding amount or number of projects that will be funded under each award mechanism for the March 14th or other deadlines? And right now the answer is no. We're actually waiting to see what we get. So we actually don't. Again, it has to be a reasonable amount that you're asking for. And hopefully you know also that if you are at 500,000 or above for any 1 year, we do have to have an extra level of approval for that. If we have the next question is, if we have a preliminary biomarker that shows someone has epilepsy and is going to have a seizure, can this mechanism be used to get more clinical data to optimize the biomarker? Right, so here I guess the guidance would be make sure that you have fairly good analytical validation in place that you can put into your application as preliminary data, and also show the preliminary data suggesting that this is a reasonable biomarker. If those things are met, then yes, you can use the clinical validation FOA to further validate the biomarker. And we do understand that while you're doing validation, you may be continuing to optimize the biomarker. Okay. Okay, we did the vision already. So would translation of the measurement of a biomarker from a low throughput platform to a high throughput technology be supported? That one's a little bit more, I guess we'd have to ask the question, are you validating the low throughput to high throughput? So everything has to do with validation. If validation is not part of it, then these funding opportunities would not support what you're suggesting. Okay, looks like the final question we have is, the milestone for U01 has to be necessarily on a yearly basis. Is there flexibility on the go, no-go time point depending on the scope of the project? If you're meeting in between years, we actually would like to see it on a yearly basis. If you're asking a question more about the overall number of years that you need, there is flexibility there. But again, I'm not exactly sure if I'm answering your question the way you had intended, so if you can email me as well, or we could set up a call on that one. Then another question has just come up for the epilepsy biomarker. Would this still be PAR 18549 analytical validation? So only if it was to validate your measure, in an analytical fashion. If you're trying to do clinical validation of any sort, it would not be that funding opportunity. Okay. Okay, so in terms of letter of interest for everybody else who's still thinking of questions, letter of interest is not a requirement, but it is something we would like you to send in, and today or very soon would be a good time to send it in. It is going to be helping us, especially for purposes of review, it'll help us figure out how many applications we're getting in for each funding opportunity, so it really does help us a lot. All right.
And I've also had a couple of questions about whether or not these slides will be made available. Yes, they will be made available to everyone that has registered for the webinar. I will be sending them out as well as a recording of the webinar. There are certain things that we have to do on our end to make sure that the audio is it can be sent out. So I would expect that to be definitely by the end of, I'd say by the end of this week, you should be getting that. If you do not, please send an email to myself. My email address is timothy.lydon@nih.gov. That's T-I, M as in Mary, O-T-H-Y .L-Y, D as in dog, E, N as in Nancy, nih.gov. Seems to have gotten to the end of the questions. We're going to kind of hang out here.
So, Tim, it looks like we have 2 more that came to Victoria. Let me just answer 2 that were sent to Victoria. One question was whether or not genetic or epigenetic biomarkers were in scope, and the answer is yes, they both are. Another question was whether or not biomarkers related to rare disease only was in scope, or whether it applied to all neurological diseases, and the answer is it applies to all neurological disease.
Okay, we're going to kind of hang out here for like 1 minute to give people, if they want to, have any last-second questions. So if you do, please feel free to send.
Okay. Looks like another question just came up. Can this mechanism be used to validate a rapid biomarker test on a disposable device? And as far as I know, the answer would be yes. Is drug efficacy monitoring a biomarker of interest? And the answer is yes. Again, we're completely agnostic when it comes to the type of biomarker. It just has to be a biomarker. I should emphasize, we are not talking about clinical outcome assessments or surrogate endpoints, so it has to be a biomarker.
All right, I think we're going to end the webinar. Like I said, please reach out to us if you have any questions. We're always looking for feedback about how we did, and if you have any suggestions on how we can make this more informative and better for you. Like I said, I will be sending out the slides hopefully by the end of this week. So with that, I hope everyone has a very happy Valentine's Day and takes care.